In today's lecture, we will talk about the angiosperms, which are the flowering plants. We will start by talking about new traits of angiosperms, including flowers and double fertilization. We will go over parts of the flower. We've done this before, but we will do it in more depth this time with a few new terms. We will also cover development of ovules and pollen, and we will talk about development of both seeds and fruits. This is the last major group of terrestrial plants for us to talk about. This diagram or sketch you've seen before, and these are all of the different major groups of plants. We start with the charophytes, which of course are algae, not true plants, but they gave rise to all of these other plant groups. We talked about the bryophytes, the lycophytes, the ferns, and then we said there were two groups of seed plants, the gymnosperms with naked seeds and the angiosperms with clothed seeds. And we will talk about why that is near the end of today's lecture. This slide shows those same major groups of plants, but on the phylogeny, so we can be reminded of their relationships. Remember that all of the terrestrial plants evolved from a charophyte ancestor. So the charophytes are shown here. Then we had the liverworts, mosses, and hornworts all branch off. Those were the groups that had a dominant gametophyte generation. Then we have the groups with a dominant sporophyte generation, the lycophytes as well as the ferns. And in each of those cases, remember the gametophyte is a free-living generation. Then we said with the seed plants, the female gametophyte is now attached to and dependent upon the sporophyte for its nutrition. And that is unique to the two members of the seed plants, the gymnosperms and the angiosperms. We talked about the gymnosperms in the last lecture, and so here we are out on this tip with our last group, the angiosperms. Angiosperms are incredibly important both to humans and in an ecological and evolutionary sense. To give you some context about this, the three groups of bryophytes, the lycophytes, the ferns, and the gymnosperms all together have somewhere around 30,000 species as an estimate. In comparison, angiosperms, that one group alone, probably has more than 300,000 species. So we are talking about 10 times as many angiosperm species as all of the other plant species combined. So something has happened here to make angiosperms incredibly successful. We will talk about the fact that that probably relates to flowers and fruits. Angiosperms are the dominant plants in most biomes. So whether you go to the Arctic tundra or to temperate forests or even to deserts, most plants that you encounter will be angiosperms. These are also by far the most important source of food for humans. If you go through all of the plant material you eat in a day, I'm guessing that more than 95% of it, maybe more than 99% of it, is angiosperm plant material. And one of the key new features of angiosperms is flowers. As we said in an earlier lecture, a flower is a specialized organ that facilitates pollination and protects seeds during development. So perhaps flowers relate to the fact that angiosperms are so successful. Why then are flowers so important? Basically what they are doing is allowing for an efficient exchange of gametes by pollinators. If you think about how gymnosperms are pollinated, they do make pollen, but the pollen in almost all cases is dispersed by the wind. That means that when the plant releases its pollen, there's an exceedingly small chance it's going to reach an ovule on another individual of the same species. Most of it's going to blow away and end up, let's say, in a lake or on the ground, or perhaps all over the windshield of our cars. So if you live in an area with a lot of pine trees, you see that there's a thick yellow coat of pollen during some seasons on every surface. That's because the trees are so bad at directing their pollen specifically towards an ovule. Wind just goes in any direction. In contrast with angiosperm flowers, most of them attract pollinators. And a pollinator is going to be some sort of animal that moves from flower to flower. Flowers incentivize visits by pollinators by making nectar, which is a sugary liquid, as well as by making pollen, which can act as a food source. Flowers, you've probably noticed, have a variety of shapes and colors. They're interesting to look at as a result, but one reason that flowers do this is to encourage pollinators to keep going to members of the same species. So when an angiosperm makes pollen, in most cases, instead of sending it out in the wind to be dispersed, instead it gets picked up by a pollinator. It is easier for pollinators to go from flower to flower if those flowers are the same shape and color. So that pollinator, when it leaves, is likely to go to another flower of the same species. And that means the pollen is going to be directed very specifically towards a place where it can encounter ovules. So because the angiosperms are much more specific in being able to spread pollen, they are more successful and they've had more opportunities to evolve lots of different species. So let's review the parts of the flower. As we said earlier, there's a stem that comes up to the flower. This is called the pedicel. There's also some base of the flower, which is labeled here as the receptacle. We talked about that before, but we're going to add a new term here that you may have encountered in laboratory already. This new term is the Pantheon. Most flowers don't make this, but many angiosperm flowers do. And this is when the receptacle, instead of being one spot down here, it gets modified into a cup shape. So this green part that I am going over with my mouse is all the receptacle. Or it is the receptacle, but when it's in the cup shape, it has a new name. That new name is the Pantheon. This is a useful trait when we're identifying flowers, because some flowers have them and some flowers don't. You can see that many of the flower parts, like the sepals, the petals, and the anthers, on this flower, they're attaching at the top of the Pantheon rather than down here at the base of the flower, as we've seen in other diagrams. So the next two parts of the flower are going to be the sepals and the petals. The sepals are outermost. In most species, they are green and protective. And what are they protecting from? Especially during flower development, before it's opened, they are protecting the flower from getting bacteria infections or fungal infections, or from insects that come and might want to eat that flower. In most flowers, then, the next layer in are the petals. And usually the petals are bright and attractive. They're yellow and large on this particular example. And so those are going to be used to attract pollinators. Now we're going to add in here a new term that you may have seen already in laboratory, and this is when the sepals and petals look very similar to one another. So the petals still look like normal petals. Those are the three larger ones in this photograph. But you can see that the sepals, which are the three smaller ones back here, in this case, instead of being green and protective, they are brightly colored and attractive. In other words, they look like petals, or we could use the word petaloid. Either way, since the petals and sepals are similar, and they are serving a similar function, we're going to give them a new name in this case, and we can refer to them all as tepals. We are going to move on now to the next layer in the flower, and this is review again. This next layer is the stamen. Remember that the stamen is used for pollen production, and it has two parts. The important part is the anther, and the anther refers to the set of pouches up at the top where pollen is produced. So this area up here. There's also, in most cases, a filament, which is a thin stalk-like structure that's going to hold the pollen up so it is more likely to encounter the pollinator when a pollinator flies in. We're going to talk a little bit more about the structure of the anther than we have in the past. Most anthers have 4 sacs, and so here's our anther here. Imagine that we took it and we cut it crosswise like this. If you made that cross, made a thin section of it, and then laid it down on a microscope slide, this is what you would see. So each one of these is really a cylinder spanning the length of the anther. And you can see there's 4 openings, and there's little red dots inside, which represent the pollen grains. Initially, what's formed inside each of these sacs, it's going to become a pollen grain, but it needs to develop to that point. So the initial thing produced are male spores. Remember that spores are always made by meiosis, and so spores are always haploid. Once formed, then those spores are going to divide by mitosis to make haploid male gametophytes. Remember, a gametophyte is always a multicellular structure or a multicellular organism. So we have several cells, or at least a couple of cells, that result in this gametophyte. And that gametophyte then gets covered by a thick outer wall. That wall is going to protect it from drying out, it's going to protect it from being eaten easily, and it's going to help it as the pollen disperses. The gametophyte with the thick wall together is referred to as a pollen grain, or just as pollen. Pollen, if you look at it close up, is super cool looking and very distinctive. In pollen grains, there are pores out of which pollen tubes can grow. We'll see pollen tubes in a little bit, but you can see, especially on these grains, there are easily seen openings, also here and here, where pollen tubes are going to be able to grow out and eventually carry sperm to the ovules. So why are there so many different pollen shapes? The shape of the wall can affect wind dispersal because there are some kinds of flowering plants that still use wind to disperse, or the shapes can help the pollen stick to a pollinator or stick to the stigma when it eventually lands. So pollen is going to land on the female part of the flower, which is the carpel. And we've talked about the basic structure of a carpel before. It has 3 parts. It has the stigmas at the very top, where pollen lands and germinates. When that pollen germinates, it's going to send a pollen tube down, and it's going to grow through the passageway called the style. So that's pictured here. And then that pollen is going to eventually reach the ovary. And in the ovary, it's going to fertilize an ovule. We will look at that. So that's review. We're going to add just one term in here. Sometimes a carpel is actually made out, or excuse me, I misspoke. Sometimes multiple carpels are fused together. When they are, they form a structure called a pistil. So we'll use the word pistil if either we have just one carpel by itself, not fused to anything, or for the joint structure if we have more than one carpel fused together. For example, in this diagram, we've been referring to this entire structure as a carpel, but that's really a bit of an oversimplification. You can see down here at the bottom there are multiple sections. We can see 2 in this picture. But if we look up to the top here, we can see on the stigma that there are 3 different lobes. Here's one lobe, here's one lobe, and here's one lobe. So if there's multiple parts down here and 3 lobes on the stigma, that tells us that this pistil is actually formed out of probably 3 different carpels. So the 3 different carpels are attached to each other on this middle seam. There's probably one in the back here that we can't see because it's behind these 2, and each one has its own stigma lobe. The 3 of them together are the pistil, and each one of the subsections is a carpel. We're going to move on then from the structure of a flower to the development of the ovules that are inside the flower. Remember that the ovules form in the ovary. And here you can see there's one ovary, but it has, in this picture, about 6 different ovules that are visible. So we can have more than one ovule per ovary. In the ovule, we have some new structures that are different than the gymnosperm ovule. So there will be many similarities, but we're going to focus here on the differences. You remember in the gymnosperm ovule, there was a protective coating around the outside, and that was referred to as an integument. Well, we still have an integument in the angiosperm ovule, but now you can see there's not just one of them, there's actually 2 of them. And so we call this the double integument. There is a stalk-like structure that we didn't see in the gymnosperm. This is going to be where the ovule attaches to the wall of the ovary. And the tissue in the ovary that it attaches to is called the placenta. This word is the same word we would use in animals where the fetus is attached to the mother, because here this is analogous to the fetus. It's the next generation. It attaches to the mother, and so it's going to attach on the placenta. And the placenta in this diagram would be along this wall right here, where each of the ovules is attaching to this ovary. So early in development of the ovule, we are going to have only tissue made from the parent sporophyte. So this initial structure is all diploid. It's all made by the mother plant. That mother plant is going to make one large cell that is going to divide by meiosis. So remember, the mother plant is diploid, but when this cell divides by meiosis, it is going to make 4 daughter cells, and each of those daughter cells is going to be haploid. So 1, 2, 3, 4 cells, but you see one is very big, the other 3 are really small. These 3 cells are going to die, and we're not going to talk about them anymore. We're going to focus instead on this big one. This big one is going to be our spore cell. So remember, the spore is going to be haploid, and it is going to divide by mitosis to make a female gametophyte. This same process happened in the gymnosperm. We still had one haploid cell that divided by mitosis and made a multicellular female gametophyte. Here, that's going to happen, but it's a more specialized female gametophyte. It is made up of 7 cells. I said 8 here, but that's a mistake. And each of those 7 cells is going to have some role. We are going to focus on the 2 most important of them. They are the egg cell, which is down here near the openings of the integument, and also this central cell. There's something strange about this central cell. First, it's quite large, but second, it doesn't have one nucleus. Instead, it has 2 nuclei, pictured here and here. The egg cell is a normal cell with just 1 nucleus. So we've now talked about the development of the female gametophyte. This is our female gametophyte. It is haploid, and it is multicellular. The next thing that's going to need to happen is the egg that the female gametophyte has made is going to need to be fertilized. And we'll see that, unlike a gymnosperm, this central cell is going to be fertilized too. So how does this happen? Well, we've said that the pollen grain is going to land. It will land up here at the top of the stigma. Then it will grow downwards, and it will enter the ovary. Once inside the ovary, it will find an ovule. What does that look like close up? Somewhere up here at the top of the screen is the top of the ovary. And this pollen tube has grown down. It has found the opening through the two integuments, and it has entered inside the ovule. Once it does this, it's going to deposit 2 sperm. Those sperm are then going to get some help to move to their appropriate locations. And what we're going to see is something called double fertilization. What does that mean? One of the sperm is going to donate its nucleus to the egg cell, shown right here, and that's going to merge with the nucleus of the egg cell itself, just shown just a little bit behind it, and that's going to create a diploid zygote. So just like all of the other cases we've talked about, a zygote is one cell resulting from fertilization. It is diploid, and it can divide by mitosis to make a sporophyte. That's the fate of one of these sperm nuclei. But the other sperm nuclei is going to make its way all the way up to the central cell, and then it's going to combine with the 2 nuclei that were already in the central cell. So now we have, instead of just 1 copy of the DNA, or 2 copies, we have 3 different nuclei contributing copies of the DNA to 1 fused nucleus. The result is that this cell is going to have a very unusual arrangement of DNA. It is going to be 3N. It's going to have 3 copies of every chromosome rather than 1 or 2. And when this cell divides, it's going to create a special kind of tissue that we have not seen before called endosperm. And endosperm is going to be nutritional tissue that the zygote and then the developing embryo is going to use as its source of nutrition as it starts to divide. And so let's look forward in the process to what's going to occur. The endosperm is dividing by mitosis and creating many cells. You can see these dashed, or these grid lines are supposed to represent multiple cells, probably hundreds or thousands of them. And every one of these cells is going to be triploid, or it's going to have 3N, 3 copies of each chromosome. Meanwhile, the embryo is going to be dividing by mitosis, and so you can see this also is a multicellular structure. And it is going to grow up and into the endosperm so it can derive nutrition from the endosperm. The embryo was only fertilized once, and it started with 1 copy of the chromosomes. That means it now has 2 copies. It's 2N, or diploid. The embryo is going to continue developing until it starts to appear like an immature plant. And there's going to be some differences in how seeds develop between monocots and dicots. For today's lecture, we're going to really focus on the dicots, and we can come back and talk about monocot seeds when we talk more about monocots. So focusing on dicot seeds here, this is what will result. As the embryo develops, it's going to develop 2 things that look like small juvenile leaves, or embryonic leaves. Here's one of them on the left, and the paired one on the right. So this seed, in other words, is going to have 2 cotyledons. Well, if we take the root for 2 as being di, then we can refer to this embryonic plant as being a dicotyledon, 2 cotyledons. And this is where we get the word dicot from. It's just an abbreviation of dicotyledon. We'll see when we do talk about monocot seeds that they have only one of them. The prefix for one is mono, and so we're going to call them monocotyledons. In addition to the embryonic leaves or cotyledons, we're going to see a couple more structures. There's going to be an embryonic stem right here, beneath the cotyledons, and at the very base of it is going to be an area that becomes a root, and I'm just going to refer to that as the embryonic root. While the seed is maturing, the ovary around it is also maturing. So if we come back up to a diagram of the flower, remember the ovule's inside here, and it's developing into a seed. But the entire ovary around the outside here is developing into a fruit. Skip ahead a couple. And so the ovary is both going to mature and enlarge. In many cases, the ones that we think about when we think about fruit, it becomes fleshy. So something like an orange or an apple or a strawberry would all be examples of fleshy fruit. In other cases, though, we can still get the development of a fruit that's not fleshy. For example, if you look up here at this maple samara, this is something you've probably seen floating around on fall days, falling out of maple trees. And this is a specialized kind of fruit that has wings on it to help it disperse in the wind. Even though it's not fleshy, it's a mature, developed ovary, so we still call it a fruit. A coconut has this shaggy, barky layer around the outside. This is a mature ovary, so it's a fruit. And then you might see these, it's an invasive plant in South Carolina, eaten by birds, and this is a normal fleshy berry. It is also a fruit. So the ovules are maturing into seeds, the ovary is maturing into a fruit, and the fruit is doing a couple of things. It is protecting the seeds inside. You can see this is a hard outer layer here and here. It is also aiding in dispersal. In the case of the maple here, it's aiding in dispersal by helping the seeds float away in the wind. In the case of the coconut, this structure floats, and so if you picture a desert island, then if the coconut can make it down to the water, it can float away and it has some chance of landing on a different island. In the case of the Russian olive fruits down here, part of the reason they are successful invaders is because birds love to eat these. Birds eat them, and then while they're digesting, they fly away. They end up in some other field, and then they defecate out their waste. Part of that waste is going to be the seeds, and the seeds will usually make it through without being digested. So those seeds then can start to grow, and they've got a good chance of surviving because they're growing inside bird scat, which has all sorts of nutrients. So fruits are aiding both in dispersal and they are providing protection. One thing I've experienced in teaching similar courses is that students often get confused between the protection provided by integuments and the protection provided by the ovary. Those are two different things. The gymnosperm has one of them, but not the other. So let's go over this one more time. For a gymnosperm, pictured on the left here, you can see that the ovule has an integument, but the integument's not sitting inside of anything. Instead, it's just sitting on top of a scale in a cone. This is why we call the gymnosperms naked seeds. The seed is not enclosed by an ovary. In contrast, for the angiosperm over here, then we still have the ovule, it still has an integument. It happens to have two now instead of one. But what I want you to focus on is that this ovule is sitting inside the ovary wall, and the ovary wall is providing a second layer of protection. In this diagram, there's only one ovule, and that's just to make the diagram easier to draw. But in reality, An ovary usually surrounds multiple ovules. And we can see that if we look back at any of our flower illustrations. In this ovary, there were about 6 ovules, not just 1. So, because the angiosperm is enclosed, we call it a clothed seed, and that is the root that gives us the word angiosperm. We are going to end with some final thoughts. First, flowering plants dominate terrestrial environments, and something about their flowers and their fruits makes them extra successful, especially this ability to disperse. Second, so we just said this, flowers and fruits are important to their success. Next, I want to point out that almost every plant-based food product is an angiosperm product. I said this earlier, but some examples. Corn, wheat, and rice are all from monocots. Next, the vegetables and fruits that we eat are mostly dicots. There are some monocots as well, but there's very few gymnosperms or other plant types. And if you eat nuts or seeds, then almonds or walnuts or sunflower seeds, those are all dicot products too. Finally, we've been really focusing on plants because this is a botany course, but we should also think about the fact that when flowers evolved, insects were evolving with them. We have many, many species of insects, and part of the reason for that is that there were many different kinds of flowers that could be pollinated, and the many different kinds of angiosperms provided many different food products that different kinds of insects could eat. So flowers are reliant on pollinators, but flowers also have helped contribute to the diversity of pollinators, including bees and butterflies. They've also helped contribute to the diversity of other insects that eat plants. When we move on to our next lecture, we're going to still talk about angiosperms, but we're going to talk about some different groups within the angiosperms.